Okay, frickin' frack.
Flipping.
Now listen,
Bunch.
we're going to talk about the next of the biomolecules. We've already started with carbohydrates. We did, what else did we do? Lipids.
We have to do nucleic acids.
We're going to do nucleic acids, then we're going to do proteins. Now we only spent about a day on each of those because they weren't that much to talk about. I mean, there obviously is a lot if I was going to do some biochemistry, but we're not giving that much. I'm going to spend most of my time on proteins, which is coming up tomorrow. But today, before we hit proteins, I'm going to give you a worksheet, which is a lot of the pictures that I've been kind of showing you that are in your papers. And I'm going to have you answer some questions. And I got to tell you, I created the ones from this morning's class, and a lot of them were, there weren't a lot of 20s out of 20. Let's just put it that way. So you're going to have to pay attention here. You get to use your notes, but you're doing them on your own. You're not working together. And you want to get this done before the end of the class, because the other class did, but they had a double period, obviously. All right, nucleic acids, what are they? Well, they're composed of 3 parts. And you guys know that as soon as I start talking about it, you're probably going to recognize it as what makes you up. Okay? And basically it's composed of 3 parts. A phosphate group, a sugar group, not quite getting there yet, but then a nitrogenous base. And those nitrogenous bases are adenine, thymine, guanine, cytosine. So what does that remind you of? What is a combination of nucleic acids called makes you what you are?
DNA.
DNA, yeah, it's just deoxyribonucleic acid. The sugar group is deoxyribose. There it is. That should be a hint as to DNA. The nucleic acid, the nitrogenous base, that's going to be different for each one. The phosphate group, that also forms part of their backbone. All right, so let's talk about what these guys' role is. And first we'll see a picture. Now this is kind of a picture, I have some pictures that are like this. It's not really what they look like. It's not a big circle over here and a pentagon here, but there's a reason it looks like that. There is a pentose sugar. That means 5 pentose, 5 carbons in the sugar, 5 ring. And then some of them contain the nitrogenous base that has, well, they're different sizes, and we'll see them in a minute. But a more accurate description would look something like this. This is your phosphate group. This is your sugar. Oops. Sorry. And of course, I'm not going to make you memorize this. And the only difference here to nitrogenous base is basically the R group. Whatever that R just means a different number of carbons arranged differently. So we usually just put an R there. But anyway, all right. You guys are all familiar with these. You've all heard of them, right? Adenine, thymine, guanine, cytosine. You should remember that from your bio class, okay? Yeah. Those guys, the only difference between adenine, thymine, guanine, and cytosine is the base, the nitrogenous base. That's this guy over, what's listed here. The phosphate group's still there. The sugar is still the same. What's different is this guy, okay? The purines have a double ring. That's these guys over here. Shorter word, bigger structure. The pyrimidines, cytosine and thymine, have a single ring. Okay? And I'm going to ask you that in the worksheet. I'm going to ask you that on the test. You should know which are which. I'm not going into a huge amount of detail here, people. If you look at your own book, which I'm not even asking you questions out of the book, I'm giving you all worksheets for this. If you're asking questions out of the book, it's asking you to have a lot more detail than I'm making you know. It's having you draw structures for these guys, being able to tell aldose and ketose. I haven't done that until the Chem 2 class, so I'm not making you do that.
Because this is biology, not chemistry.
This is right, exactly.
So we shouldn't have to.
There you go. But anyway, all right. So that's what's different about that guy. All right, so if you look at DNA structure, you probably remember a few things about what DNA looks like, right? What kind of the structure? What do we always describe it as?
Double helix.
Exactly. Double helix. Very good. A double helix, double strand of nucleic acids. Very good. Stone's on fire.
You better
Put that fire out.
with the extinguisher. I think you should spray some out with the extinguisher.
Good. Very good. All right. So it's a double strand of nucleic acid. It has a double helix shape. And it is what stores genetic information. How it stores it is by that code of nitrogenous bases. Depending on the order that they're in, will determine why you have blue eyes or green eyes or brown hair. It determines your skin color, why you're a certain height, weight, everything that makes you you. Everything that makes you you is determined by your DNA. Because in small packets of information like A, adenine, thymine, just in an order, a certain order, that small piece of that codes for a specific thing about you. What do we call that part of the DNA that codes for something specific about you? Yeah, a gene. Exactly. All right, that's what a gene is. So the DNA, you have this picture on here, is a double helix. But by the way, we're going to see this tomorrow when I talk about how you actually take this stuff and make proteins from it, unzips. And I'm going to talk about that. But for now, you should know this is a double strand. You know what? That's not the only guy that uses nucleic acids. What about RNA? Ribonucleic acid. That's a single strand. It's not a double. Okay? And of course, there's a couple of differences for ribonucleic acid, for RNA versus DNA. DNA has some different nitrogenous bases and has a different sugar. Okay, we'll talk about that in a second. But the RNA, what it does, there's a couple of kinds of RNA we're going to talk about tomorrow. That one we are going to get into a good amount of detail. I am going to do a fair amount of detail for the process of protein formation, even though I'm not going into a whole lot of detail on these other guys. Because I think that's probably the most important thing. How you get from DNA to you. Okay, how it get from DNA to what makes you you. And RNA has got a vital role in that. It's a single strand. It's this guy like drawn in your picture here who comes in and matches up with that DNA and then goes off into the cell and makes the protein. And again, that's the process I want to talk about. That's going to take me 2 days. 1 day for each of these other ones, 2 days for that. Practice worksheets, one today, other ones next week, or not next week, but next couple of days, and we'll have a test by Friday. Then we're done. Okay. By the way, this same graphic is somewhere on your worksheet. You're going to get in a few minutes, so watch and listen to the kind of questions I might ask you. All right, if you see, everybody, both DNA and RNA, both of them have adenine, guanine, and cytosine in them. However, DNA only has thymine. It does not have uracil. Like RNA only has uracil. It does not have thymine. Okay? So you should know that. The difference between DNA and RNA. DNA has deoxyribose as its sugar. And RNA has just ribose, which is where the words come from. Ribonucleic acid, deoxyribonucleic acid. And the only difference really is a OH group, basically, over there. He has one where this guy doesn't. Not a big deal. But you'll be able to look at your notes for the worksheet. Okay? Eventually, you're going to have to know some of these things for the test, but I will let you know exactly what you need to memorize before the test comes around. For today, you'll get to use your notes. Okay. All right, so the other thing about DNA I need to talk to you about is when its structure is made. Here it is. Again, let's take a look at these again. You have this picture, I believe, too. Do you or no?
Yeah.
Okay. These bonds here that make up the backbone of the ladder, okay, they're all, this is a phosphate group, the sugar, and then the nitrogenous base. Notice how the bases pair up. A always goes with T, and G always goes with C in a DNA molecule. Okay? And if you notice, that makes sense, because otherwise the ladder would be twisted. If I didn't have a double, like a bigger one and a smaller one, with a bigger one and a smaller one, if I had two bigger ones or two smaller ones, the ladder would be twisted. Okay? So that's the only way they pair up. And they pair up with weak hydrogen bonds. Hydrogen bonds, we said, were the strongest of the intermolecular forces, but they're weak compared to this. So this guy's weak enough to zip open, which you'll see why it has to zip open tomorrow. Okay? So the nucleic acids are linked in this way. The backbone of the ladder is linked by covalent bonds, like I was just telling you. In other words, the phosphate and the sugar are all hooked up down this way. All right? And they're all hooked up with strong covalent bonds that don't break. Well, you hope they don't break. When they do break, it could cause a mutation. And that can happen from chemicals can do it, radiation can do it. That causes damage. It could kill the cell, or it can cause cancer. So we don't want that to happen. We want these bonds to stay where they are. However, we want these bonds to be able to unzip and break. And that's why these bonds that hold one ladder rung to the other, is basically held by hydrogen bonds, which, again, are strong compared to intermolecular forces, but weak compared to covalent or ionic bonds. And again, you'll see the purpose of this tomorrow when we talk about protein synthesis.
You get a grade or a test?
I already have my set to record into the thing right now. I'll do that actually while you're working on your papers. All right. How do they pair up? As you can see, you're going to have to know this sequence. They only pair up A and T, A with T, and guanine with cytosine. Okay, I think I have that written already in there for you, right? Okay. All right. So I try to save you some time. That's why we got this papers. We only have a little bit more to get here today, and then you'll be able to finish that worksheet, even though I spent a little bit more time with the other class. All right. Who are those two guys? Don't look down. I didn't look down. Yeah, well, it's already on your paper.
This literally looks like you took it from Raw 3's.
Yeah, it may very well be the same one. It may very well be the same.
No, I mean like the whole thing. Looks like you took it from
No,
Raw
I
3.
did not. She took it from.
Looks like.
It actually may be the same, but I did this myself, and actually she may actually have the same exact stuff, but I would have given it to her, not the other way around. I made it up. Actually, I didn't make it up. Let's face it, I stole all this stuff.
Yeah,
it's all plagiarized from the internet. Actually, some of it's really not plagiarism. Like a lot of those graphics you'll see are from when we buy our textbooks for the district. They come with all the stuff loaded on a CD or a DVD, where I'm allowed to use any of that stuff.
You're allowed
What's
to play
the girl that we're supposed to shout out to that also came up with this? I forget her name.
I forget. Anyway, okay. So Watson and Crick, by the way, Watson and Crick, some people would say got a little bit too much credit for what they did because they built their work on a lot of other people. But eventually they are credited with the discovery of this structure, this three-dimensional double helix, like a twisted ladder, like a spiral staircase, basically. And that structure's shape is really a big deal. Coming up with that is a big deal. It explains how, and then, by the way, as that folds, it's able to pack so much information into your genetic code. Okay? Why are you so complex? Well, it's just a four-letter code, A, D, G, and C. But basically, I can, with 4 letters, write a lot of words. Okay? And I can, and depending on how long that stretch is, maybe 100 or 50 or whatever, nucleic acids long, that particular stretch of DNA is basically going to code for a specific protein. And they actually are very good at knowing this. A lot of the genome of the human, actually, pretty much the whole genome of humans, has been mapped. So we know what it looks like. And we also know we don't know what every single part of the DNA codes for, obviously. But we have identified certain parts that do code for certain things. Like you could tell which part is going to code for a specific protein, like insulin. And you know where some of these things are. And that's why they're able to do the genetic modifications that they do, not in humans, although they're talking about doing that even too, but in plants. For example, I can make a fish which has genes that make it grow faster. So I could splice those genes in, create a different species of fish. I could do that with corn, so it can be resistant to certain bugs. And they're talking about, and as sick as it may sound, I just read an article the other day about basically putting human and animal DNA together, all right, basically making chimeras or hybrids of the two, so they can grow organs for humans. Like, in other words, have a pig that has a human pancreas that we could transplant from one to the other. I mean, it sounds pretty weird and horrible to me, but we already can do a lot of that stuff on a very simplified basis for certain plants and certain animals. We don't do a whole lot of that gene splicing for humans, but that may be next and probably will be. And that will be, by the way, something that in Chem 2, again, we talk about. Several people did their articles, their research papers on genetic engineering or cloning or things like that. And there's a lot of ethical debate, which we won't have time to get into here, but we will in Chem 2. All right, so there it is. That's a very impressive molecule. And remember, this is going to be incredibly long. It's not just going to be that big. It's going to be very long, making up, with all the genes that make you what you are. Okay. Now tomorrow, we're almost done. We're going to finish this last board here. Tomorrow, we're going to begin to explain this big mess here. Because that is what was called the central dogma of molecular biology. That's what it's called. And it basically says that DNA is responsible for making proteins. It is the blueprint or the map that tells you what you are supposed to look like and make in your body to keep you alive. And this is going on trillions of times every minute of every day in your body, in the trillions of cells you have. Each cell is doing what it's supposed to do to keep you alive. And it's making specific proteins and coding for those things from the DNA that's in your body. And because everybody's DNA is different, you all look different, you all act different, you all have different susceptibilities, different everything about you. Your hair, from everything from your outward appearance to pretty much everything. But that is a complicated process. We're going to talk about how it unzips. The RNA comes in here, makes a copy, goes out into the cell, hooks up with a ribosome, gets some amino acids, and it's complicated. So that's why we're going to spend more than just one period on that part. But the last thing I want to say about nucleic acids and DNA is that's the source, and it goes in one direction from DNA out to making proteins. DNA stores the information in that code, that 4-letter code, A, T, C, and G. We will talk about how that gets transferred out starting tomorrow. Because it depends on the arrangement. This is the last of the notes here. Depending on the arrangement, a certain sequence of DNA, we call that a gene, codes for a specific protein. And that's why you are what you are. That's why I'm so handsome. That's why you're so ugly. It's all in the genes. And I don't mean Levi's or Wranglers.
Thank you for clarifying.
Maybe you could add it like laughing in the video, so it sounds like
Or clapping.
yeah.
Because I'm done. All right. I'll give you that worksheet because you'll have, like I said, I went a little quicker here.